Did you know that the global human population is expected to increase more than 9.8 billion in 2050? The increment in the population required more energy and caused more toxic gases emitted. One of the main sources of the problem that may occur is usage of fossil fuel as the energy source. The use of fossil fuel has led to many critical problems that affecting society, such as global warming, emission of greenhouse gases, and air pollution. One of the most significant effects of burning fossil fuel is the gas release. All fossil fuel emit carbon dioxide and other harmful air pollutants when burned. These emissions lead to a wide variety of public health and environmental costs that are borne at the local, regional, national, and global level. These problems have affected the homeostasis of living organisms worldwide at an alarming rate. Due to this, it is important to seek for an alternative to reduce the usage of fossil fuel, and one of the relevant substitutes are biofuel. There are different types of biofuel that have been previously explored, but recently the use of microalgae has been strongly considered for the production of biofuel since they present a series of advantages over other biofuel production, such as they do not need arable land to grow and therefore do not compete with food crops compared to the first generation of biofuel produced from corn, sugarcane, and other plants, and also they exhibit rapid biomass production containing high oil content, which is 15 to 20 times higher than the land-based oleaginous crop. Hence, these unicellular photosynthetic microorganisms have received great attention from researchers to use them in the large-scale production of biofuel. Photosynthetic organisms such as microalgae convert water and carbon dioxide into the chemical energy and valuable components such as lipid, protein, and carbohydrate. In the process of photosynthesis, non-polar lipids such as triacylglycerol store in the cell. This product has high commercial value as TAG can be converted into fatty acid methyl ester, which is a fundamental component in biodiesel. Esterification is the process used to convert TAG to FAME. Efficient cultivation in large scale can produce high lipid amount to supply the biofuel globally. However, one of the disadvantages of using microalgae is the high economic cost due to the low yield of lipid content in the microalgae biomass. Thus, development of different methods to enhance microalgae biomass as well as lipid content in the microalgae cells would lead to the development of a sustainable low-cost process to produce biofuel in a shorter time. Hence, in my study, I will focusing on increasing the lipid content in various freshwater and marine microalgae species by optimizing the growth parameter, which include nitrogen, salinity, and iron content. Based on the research done, nutrient limitation can affect the metabolism, growth, and also reproduction of the microalgae. Undesirable conditions occur during nutrient depletion, force the cell to produce more lipid, and this technique was used by researchers to increase the lipid content. In high concentration of metal, reactive oxygen species produce an inhibition in the synthesis of chlorophyll happen, which resulting in the increment of lipid accumulation. Another factor that can be manipulated to increase the lipid production is salinity stress. Salinity stress changes the metabolism of microalgae due to biological mechanisms relating to growth and other physiological. It uses the osmotic mechanism to manipulate the metabolic pathway and yield more lipid production. So stress can lower the sodium-ion or hydrogen-ion antiporters activity and affect the oxygen evolving machinery and photosynthetic machinery. High sodium ion concentration lead to change in physiological condition of microalgae and resulting more accumulation of lipid that help microalgae to survive under the stress. That's all for me. Thank you.